Okay, so this is a video on the phases of mitosis and meiosis, but I'll start with mitosis first, okay. Before you actually start mitosis, so before all of the cell division, a cell goes through something called interphase, and it spends about 90% of its time doing interphase. So interphase has 3 stages or 3 parts to it. And what I'm actually going to do is I'm going to draw something for you. This is called the cell cycle, and I'm going to draw the 3 stages of interphase in the cell cycle for you. The 3 stages are called G1, S, which stands for synthesis, and G2. The 4th part that you see here is mitosis and cytokinesis, which is part of the cell cycle but is not part of interphase. So we're just going to cross that out for now because we want to focus only on interphase. So what happens during these periods? Well, G1 is a period of rapid growth. During S phase, you have the replication of chromosomes. And what that means is if you have a single-stranded chromosome, as you see here, it will duplicate itself and become a double-stranded chromosome. Okay, so let's just remove that for now and move on to G2. And G2 is just the time of more growth and preparation for cell division. And once a cell has done all that, it will be ready for mitosis. So let's just delete all that and begin with the actual explanation of mitosis. So at the end of interphase, you have something that looks like this. This is a cell, and at the center of it, you have something called chromatin. Chromatin is surrounded by a nuclear envelope. Also inside of the nuclear envelope, you have this little thing called the nucleolus, which is just a compartment that holds molecules used in DNA replication. The red things at the bottom that I'm drawing are called centrosomes, but one of them is called an aster, and an aster is made out of 2 centrioles. So a few things happen in prophase. As you can see, the nucleolus disappears, and the nuclear envelope begins to fade and dissolve. Also, the chromosomes begin to shorten and thicken, while the centrosomes move to opposite poles of the cell. In metaphase, the nuclear envelope completely dissolves and is no longer there. The chromosomes line up at the center along an imaginary line called the equatorial plate. As the centrosomes had been moving away to opposite poles, they were also connecting themselves to the chromosomes with these little microtubules, and we call them spindle fibers. In anaphase, the chromosomes split along the centromere. The centromere is just the point of connection between the 2 duplications of the chromosome. The spindle fibers pull away the halves of the chromosomes from each other. In telophase, the chromosomes have been completely pulled away from each other and now are on opposite sides. They begin to lengthen. Also, the nuclear envelope reappears. And while this is happening, you also have a process called cytokinesis happening. And cytokinesis is just the splitting of the cytoplasm of each of the cells. So now you have the formation of 2 different cells. This cytokinesis is not part of mitosis, but it does happen usually right after or even during telophase. And by this time, the nucleolus also reappears. And now you have 2 separate but identical cells, which is mitosis. So once you've done that, the 2 cells will now separately go into interphase, so back to G1, S, and G2. And once that has happened, you're at the end of interphase, the cycle will begin again, the cell will go through mitosis, it will divide, and so it goes. Okay, so now meiosis. And in meiosis you have interphase and then you have prophase 1. And in prophase 1 there's a few things that happen. So your nucleolus disappears and your nuclear envelope begins to dissolve. But there's also something else that happens, and that's called crossing over. And crossing over happens after synapsis happens, which just means that two homologous chromosomes find each other. And once they have found each other, crossing over is where they swap DNA. So as you can see, the blue part gets a little bit of the pink, and the pink part gets a little bit of the blue. And that increases genetic diversity. The location where crossing over happens is called the chiasmata. And once again, in prophase 1, the centrosomes move to opposite poles of the cell. In metaphase 1, the nuclear envelope completely disappears, and the homologous pairs line up at the center or at the equatorial plate. And once again, spindle fibers from the asters connect to the chromosomes. In anaphase 1, the spindle fibers pull away the homologous chromosomes from each other. In telophase 1, the homologous pairs are now on opposite sides, so there's a single chromosome from each homologous pair on each side. And also cytokinesis happens, so cytoplasm breaks apart, and now these two become completely separate cells. And then we go back to prophase, but this time it's prophase 2. I'm just going to focus on one of the cells that came out of telophase 1, so I'm just going to expand that and we'll look at what happens there. So the nuclear envelope is once again there. The aster is duplicated, so you now have 2 of them. And once again, the centrosomes move to opposite poles of the cell. In metaphase 2, the nuclear envelope once again completely dissolves, so it's no longer there. And the chromosomes line up along the equatorial plate. Also, spindle fibers from the asters attach themselves to the chromosomes. In anaphase 2, the chromosomes are pulled to opposite sides by the spindle fibers. In telophase 2, the single-stranded chromosomes are now on completely opposite sides and they begin to lengthen. Cytokinesis is also happening, so you're now getting 2 cells out of this division. The entire point of meiosis is to get from a diploid cell to a haploid cell, and that's what we have here, 2 haploid cells. And in the entirety of meiosis, since I only did 1 cell, you'd actually get 4 haploid cells.